Welcome friends, welcome to another video from the Chanda's Biology, and in this video we will discuss about bacterial transformation method, one of the best method to transfer the genetic material from one bacterial cell to the another bacterial cell. Now what is meant by bacterial transformation actually? It is the transfer of the naked DNA from one bacterial cell to the another bacterial cell, or we also say that the uptake of DNA by the recipient cell from the donor cell. See that, suppose this is the one of the bacterial cell, which is the donor cell, donor bacterial cell, containing its own DNA, and from this donor DNA we obtain the fragments of DNA molecules. These are the fragments of DNA. Okay, now how these fragments of DNA are obtained? These are the fragments of DNA. You can say that fragments of DNA. These fragments of DNA are obtained either by the natural cell lysis or by chemical extraction. The natural cell lysis of this bacterial cell takes place, or either by chemical extraction of this DNA we can obtain the fragments of DNA, and these fragments of DNA now spread in the environment. Okay, spread in the environment and attack on the another bacterial cell, which is called recipient cell. Suppose this is the another recipient cell having its own DNA. Now these fragments of donor DNA, okay, they are attack on the recipient cell and binds with the cell wall of the recipient cell. Means they are attacking on the cell wall of the recipient cell and attaches to the cell wall of this recipient cell. Okay. Now after the binding with the surface of this recipient cell, later on, after some time, these fragments goes inside the recipient bacterial cell. But see that in these two fragments, the one fragment is totally degraded by the nucleases enzyme in the next step. Suppose this is the next step. This is the recipient cell DNA, and here are the two fragments. These two fragments now enters inside this recipient bacterial cell. But after entering in the bacterial cell, these two fragments, the one fragment is degraded by enzyme nucleases. Okay, yeah, and hence we are remaining only one fragment. Here is only one fragment is remaining, and the another fragment is degraded by the enzyme nucleases. Okay. Now this fragment is form the complex. It binds, it incorporate with the recipient cell's DNA with the help of homologous recombination. Okay. In the next step, this fragment of donor DNA is now binds to the DNA of the recipient cell with the help of homologous recombination in the next step. See that, like this. This is the DNA of recipient cell, and this fragment is bind in the fashion of, in this fashion. This is the donor DNA and this is the whole recipient cell DNA. And this new cell is known as transformed cell. Remember that the property of recipient cell before the donor DNA binds with the recipient cell DNA is different, and the property of this transformed cell is different. Suppose these fragments of DNA having such property, this gene having some property like as antibiotic resistance, and if this gene is binds with this recipient cell DNA, the property of the recipient cell is also now changes, and in this cell is have now antibiotic resistance property. Okay, and also note another point, those DNA having the molecular weight is between the 3 lakh to the 8 million Dalton are always more preferred to transformation process. Those DNA having, I written here, those DNA having 3 lakh to 8 million Dalton molecular weight are easily transformed in this process. Okay, so this is all about the bacterial transformation process. Thank you very much. If you like the video, then press the like button and subscribe our channel for the more videos. Thank you.